Want to get caught up on the latest, greatest bioprocessing equipment and solutions without putting in travel requests, flying across the country, spending gobs of money and time getting harassed by salespeople? Of course you do. Register for Bioprocess Online's BioExpo Live, being held July 30th through August 1st. This inaugural expo is a fantastic opportunity for biopharma companies and contract manufacturers to evaluate the latest and greatest from the comfort of your desktop or mobile device. Conveniently, we've broken down the lineup into upstream solutions on July 30th, downstream solutions July 31st, and quality analytical and data solutions on August 1st. It's absolutely free to register for this series of short, digestible, and interactive sessions. Just hit the link in today's show notes to register for BioExpo Live today. In the biopharmaceutical business, delivery is at least half the battle. We make tumors go away in petri dishes all the time. And of course we do. The tumor can't escape, its cells can't multiply, the drug can't escape and go on to wreak systemic havoc. It's a tightly controlled environment. In human bodies, though, the challenges associated with getting a therapeutic molecule to its target and keeping it there, and perhaps only there, are biologic, chemical, physical, and engineering problems that, left unsolved, will never allow biotech builders to bridge that gap from academic exercise to clinical success. I'm Matt Pillar. You're listening to the Business of Biotech, and on today's show, we're talking with a man who's spent the past 4 decades working on the interface between advanced diagnostic and therapeutic products and the human patient, that interface being delivery technology. Eamonn Hobbs is president and CEO at Syncromune, a phase 1 drug/device combination company whose SyncT solid tumor therapy platform combines partial oncolysis of a tumor with infusion of a multi-target biologic drug directly into the tumor. It's a novel approach using novel T-cell science and a proprietary device to deliver the goods. On today's show, we're going to get to know Eamonn, and we're going to learn how Syncromune is building a holistic biotech business that's focused as much on delivery as it is on drugs. Eamonn, welcome to the show.
Well, thank you, Matt. It's great to be here with you today.
I appreciate you being here with me today. And this episode's not going to drop for a little while, but I especially appreciate you because I know you just came off of a very busy and successful ASCO. So I hope you had a minute to take a breath before you jumped on the Business of Biotech with me, but I'm very appreciative in any event.
Well, thanks very much. Great to be here.
Mm-hmm. As I said, hard to believe looking at you, but 40 plus years in this business, working on this interface that I referenced, the instrumentation and the mechanics that interface drug delivery and diagnostics with the patient, and more so building businesses around that interface. It's a very unique, interesting niche, I would say, that you've built a very successful career in, and I'm curious about how that all got started. I mean, I know you were a bioplastics engineering guy in your undergrad and coming out of school. At what point did you say, this is inspiring enough for me to want to go build a career around, as I said, this sort of interface between biotechnology and human beings?
Well, thanks very much. I am an old guy. I'll be 66 in next week, actually.
Well, happy birthday.
Well, thanks so much. I got into combination drug device and drug delivery innovations through an interesting path. I didn't start out thinking that that's what I wanted to do. And as a matter of fact, I didn't think that biomedical engineering, material science, or even medicine was where I wanted to go. It was very clear when I saw Neil Armstrong put his foot on the moon. I said, I want to do that. I want to be an astronaut.
Really?
And no question. And I studied real hard and did everything I could to put a path together to have a shot at getting into NASA way back when in the 70s. And I got an appointment to the Air Force Academy and ended up entering in 76, which was the first co-ed year for the Academy. So, which was a big landmark. But I thought I was on my way. I thought I was exactly where I wanted to be, when I wanted to be there, and had all the tools necessary to have a really decent shot at being an astronaut. And unfortunately, it didn't work out. I was informed that I had a progressive astigmatism in my right eye and would likely not be flight qualified by the time I graduated. So, which was devastating. I mean, all my hopes and dreams just went poof, just like that. And I had 20/20 vision at the time, didn't have these, and I had to make some really serious life decisions pretty quick. And the Air Force said, you're going to have a great career in the Air Force. Don't worry about all that flying stuff. You'll have a great career in a missile silo or pumping gas for all your pilot buddies.
Yeah.
So I did the only thing I thought was rational and would work for me, and that was eject. And I left the academy, and through really great good luck, I ended up at what was called Lowell Technological Institute at that time, and now called UMass Lowell, which had a great engineering reputation, and more importantly, would take me in the middle of freshman year, which is unusual. So I was an advanced placement student, and so I caught up quickly. And I entered as an electrical engineer because that's what they said I could get into. And my oldest brother was an electrical engineer, and my father was also, along with being a lawyer. So I said, oh, that makes sense. And then once I got in there, I saw that the coolest engineering that was at Lowell Tech was plastics engineering. And I was lucky enough to get into that, and which was an incredible break for me. And then bioplastics engineering became a creation of my own. There was no biomedical engineering at Lowell Tech back then, but they gave me the latitude to kind of make it up as I went along. So I took all the medical courses, pre-med and even grad school courses that the university offered: physiology, biochemistry, molecular biology, et cetera, and just found I loved it. And I thought, hey, maybe I'll be a physician. This is kind of my calling. It appears I'm good at this stuff, and I like the engineering a lot. My brain kind of worked that way, and I was editor of the school newspaper, so I was a writer as well, which is unusual for engineers. I have a lot of engineering buddies, and communication's never been their forte, and they'd be the first ones to admit it. But I being Irish, the gift of the gab. So I thought, well, I'm thinking this is the way to go. Another incredibly good break. I was hired by my first entrepreneur mentor in my career, Bill Cook, who's a legend in medical devices. No longer with us, unfortunately, passed away in 2011, I believe. But he hired me right out of school to come and join his company that was already very successful primarily in cardiovascular devices in Bloomington, Indiana. And I came in and I was pretty naive. I at that time, coming from the Boston area, I thought Indiana was a city in Illinois. So I was pretty young and stupid. Now I'm old and stupid, but I was pretty green back then. And I settled in finally, and Bill really took a shining to me and said, you're doing great, kid. What do you want to be when you grow up? And I said, everybody called him Bill. I said, Bill, I think I want to be a doctor. And he said, no, you don't. No, you don't. I know you, and what you do is really cool. And that is you have no trouble speaking to physicians and understanding what they need. understanding what they do and what they need. And then you have no trouble communicating that to your engineering brethren back here at the ranch.
Mm-hmm.
You're a very unique individual, and pat yourself on the back. That's a really important thing in the development process of innovations in medicine.
Yeah.
That's what you do, and that's what you really are passionate about. Of course, again, I was young and stupid and arrogant, and said, no, I want to be a doctor. So he said, oh, okay, I'll tell you what, I'll pay for you to go to med school. I'll send you to med school if you give me 5 years after graduation. And I said, done. He goes, but there's a catch. You have to go up to the chief of staff at IU Med Center, who's a dear friend of mine, who's a surgeon by the name of Don Schwarten, and he's going to put you through boot camp. You're gonna be his right-hand person until med school starts next season. And if you still want to go, you got it. And if you don't, well, you've had an incredible experience. So I did, and I didn't sleep the entire time. I learned how to sleep with my eyes open, standing up. Don Schwarten was the most amazing individual I've ever come across. Incredibly talented. And I had a crash course in medicine, what it's really like to be a very active clinical physician, a surgeon. And it turned out, when you know it, Bill Cook was right.
Yeah.
I didn't want to be a physician. Patient care is really hard. And I take my hat off to clinicians who deal with that morning, noon, and night, 7/24, 365. It is an incredible commitment to make, to be competent and then gifted in patient care. Yeah. So I came back to Bill and said, okay, you're right. That was an incredible experience, and I got it out of my system. I want to really be that interface between medicine and engineering innovation. And he said, great. He said, you're smarter than you look. Good for you.
That's an incredible story.
It was wonderful.
Yeah, and it's incredible because it sort of comes full circle where you go, oh well, I just happen to have acquired both the engineering and medicine chops, right? Background necessary to build a career around that. So
yeah, it was. I couldn't have drawn it up any better if I tried, and it really the stars aligned for me. So it was marvelous. So I continued on at Cook Incorporated, and Cook was an entrepreneurial venture, and Bill was hitting it out of the park. And today, Cook Incorporated is the largest private medical device company in the world, with billions in revenue, and still family owned. Carl Cook, Bill's only child, is running the show. And Carl and I just caught up a few months ago, and was really phenomenal to see him. He was my summer intern in 1980. So
really?
Yeah, we both come a long way. He's a great guy and very passionate, as you would imagine. So marvelous. But I, again, in the young and stupid category, I looked at Bill Cook and said, hey, this entrepreneur stuff seems pretty simple. I think I can do it. So I decided to leave Cook and go off and start my first medical device business called Hobbs Medical. Learned a heck of a lot. I learned that entrepreneurship is really hard. Mm-hmm. I was a young engineer with a young family, and I had to give up an incredible, steady growth opportunity job with Cook to start my own business and live hand to mouth. And my wife, God bless her, stuck with me, and we got the business off the ground and ended up having a good exit with it. I learned about VC, and I learned about how to position a company for an exit. And I learned about, we were commercial, we were selling devices, and it was all good. One of the companies that bid for Hobbs Medical was a drug company called EZM. And that really was my transition into the world of pharmaceuticals. And EZM was a world leader in GI contrast media and devices to deliver it. So barium enemas and the like.
Mm-hmm.
Howard Stern, my second entrepreneur that I got to work for and was a phenomenal mentor, took me under his wing, and I got a real bellyful of what the combination drug device business is like from a contrast media and delivery device perspective. And back then it was really nascent. It was so nascent, in fact, that GI contrast medias were regulated as devices, not as drugs yet. And when about the day I joined the company, it seemed the FDA made a landmark decision to recategorize GI contrast medias as pharmaceuticals. So I went through the whole transition with EZM from device to drug in a drug device company. Again, incredible experience and painful experience for the organization and for the whole team.
Yeah.
But I got a real ground up education in what it was all about now to develop drugs and to develop drug device combinations. So did well there, and I was I'll skip ahead to founding AngioDynamics, but I decided I was going to start another business, and I got EZM to my buddies Howard Stern and Phil Myers, the founders of EZM, to help finance it. I started AngioDynamics with on the back of an envelope, and it was a hard to believe drug device combination company. Our first two products were that we were developing were a system called Pulse Spray, which was a partnership with Abbott on their abokinase, their urokinase product, which provided an intravascular catheter to deliver the lytic enzyme into the blood clots directly. And this reduced systemic exposure of the drug, which could lead to strokes and other very bad things, and also decreased the lysis time dramatically. So here was a perfect example of how you can take a really great drug, but make it way better with a delivery system and make it safer, make it more effective, and just add all kinds of value without having to develop a brand new drug.
Yeah.
So added a whole new life to abokinase. The other product we were developing was carbon dioxide angiography, using carbon dioxide gas as an angiographic contrast media, an intravascular contrast media. Wild at that stage. Mm-hmm. And at that time, medical gases, including CO2, were regulated as devices. So we and had very broad labeling. Basically, the indications were use CO2 whenever it's appropriate. So it was a pretty broad label to fit in. So it all looked good. So we developed Pulse Spray, had a great run with it at AngioDynamics, and developed CO2. We got our 510K, had a great partnership with Abbott on the Pulse Spray. On CO2, not so fast. We developed a proprietary injection system, really an angiographic injector that was built for compressible fluids like CO2, which would displace the blood in the area of interest, creating a negative contrast zone, and filed that 510K as an angiographic injector with the agency, the FDA, and went through a number of cycles of questions, and got a phone call from the project manager at the agency who said, "Hey, congratulations! I just sent your 510K upstairs for signature, final signature. You should get your 510K notification, in a week or two. Great job, guys." And that was an odd one. Good luck with it. And we were popping corks and thinking everything was great until we got a call from that same project manager who said, "Sorry, this got kicked upstairs for some reason, and it's on hold. Your 510K has been put in escrow," which I was thinking, well, what does that mean? Never heard of that before.
Yeah.
So ended up that David Kessler, the FDA commissioner at the time, and his team had decided that medical gases were going to be recategorized as pharmaceuticals.
Mm-hmm.
So our delivery system was no longer associated with delivering a medical gas device. It was now associated with delivering a drug that was not labeled for that as a contrast media. So we had meetings. I got to meet David Kessler many times because we were kind of a landmark precedent in combination products.
Yeah, that's what I was going to ask. I mean, this was new stuff at the time. This was new stuff for the FDA to consider.
It was. This was 1988, 89. Mm-hmm. So very new for the agency. The combination drug device pathway was still very, very new, which, we've come a long way. Last year, over 400, I believe, combination drug device products were approved by the FDA. So it's a well-paved road now, but back then it was the Wild West. We were making it up as we went along.
Yeah.
So the CO2 ended up needing to be developed as a drug, and it was an unfortunate reality that commercially no one was going to pay pharmaceutical margins on a gas that you can get in virtually unlimited supply. I mean, I'm producing a lot of CO2 right now for nothing. Mm-hmm. Nobody's going to pay pharmaceutical margins prices for that. So commercially, the project became a non-starter, and we negotiated with the agency, and we ended up coming to the realization we had to come up with another way of delivering CO2 that could fit in the device world, which we ended up doing a hand system that got by, but. Everywhere outside the United States, our CO2 system was commercially successful. Mm-hmm. So we sold hundreds and hundreds of those systems, and CO2 angiography is a kind of a standard of care these days. In the U.S., it's still done by hand, but in the rest of the world, it's done with a combination drug device system.
Yeah.
So that got me very, very well cemented into drug device kind of being my thing, and more opportunities started to come around. And I took my team and I took AngioDynamics public in 2004. We had a great run. We were the best performing medtech IPO that year, and we were Forbes top 200 growth company. We were a Business Week top 50 hot growth company, and we were just crushing it. I ran it as a public company for 5 years, which is about the average for public company CEOs shelf life, and then was promoted to co-chairman in 2009, which is code for you're out of here. So the board thought it was the time to bring in a Fortune 500 level CEO to really drive the company. It was an extremely successful company. On my watch, we got it to over $1 billion in market cap, and it was a great return for the shareholders, great return for my original investors at EZM, and life was very, very good. So that was 2009, and I had to decide what I was going to do with myself. So I decided I'd join a bunch of boards and try and give back, frankly, to young entrepreneurs. And one of the companies I joined was Delcath, that had a very interesting combination product system in oncology. And many of the products at AngioDynamics ended up being in oncology because the interventional radiology community was migrating that way, minimally invasive, focal oncologic therapies. So I had a good base in oncology, and the Delcath data, clinical data. They were phase 3, developing a ocular melanoma mets to the liver new therapy, which was a drug device combination where the vasculature of the liver was isolated, and the liver was exposed to very, very high concentrations of melphalan, one of the original cytotoxic chemotherapeutics. Mm-hmm. And it would kill the cancer, and amazingly, the healthy hepatocytes, the healthy liver cells, were virtually immune to the chemotherapy. And when you think about it, the liver filters poisons out of our blood, so it made sense that the hepatocytes be able to tolerate this. If the chemo got out of the liver, it would decimate the bone marrow, and lead to death, or at least neutropenia or thrombocytopenia, or both, grade 3, 4, really, really toxic. So it was very important that the device do its thing. And it involved a high flow blood filtration system that would filter the chemo out without damaging the blood, which had never been done before. So it was a really intriguing project and required a lot of innovation. And these patients were terminal without an innovation. So it was incredibly gratifying to be able to add, significant quality of life to these patients' lives where they really had nothing that was working before.
Yeah.
So a belly full of that, we published our interim pivotal phase 3 data. It showed incredible efficacy, and the company's market cap went to well over $1 billion, about 1.4 billion. It was 28 million when I joined the company. And we thought we were cooking with gas. We filed our NDA for the combination product and got a complete response letter, which was a bit shocking, where they said, "Hey, we love your data. It's really great, but you changed the device during the phase 3 trial. And we want you to repeat the study with the same device. We also, your study design included a crossover provision that allowed patients who failed the standard of care, which they all failed, to cross over and get the therapy if they still qualified, which was ethically, a very reasonable thing to do. But unfortunately, on an intent to treat basis, that meant the Kaplan-Meier curves were overlapping because the patients that crossed over did just as well. So there was no difference between the control arm and the treatment arm when you accounted for the crossover. When you subtracted the crossover compared to historicals, there were, first off, all the crossover control arm failed to get there. Two, if you compared it to any historical data, all the patients died. So it seemed really harsh that the agency was throwing the crossover provision in our face. But during the course of the execution of that trial, the FDA had made a big shift and said the crossovers really, although they're ethically attractive, they really do a disservice to getting the new therapy approved. You really need clean data. So you've got to randomize people and not cross them over. So we suffered through that. Delcath, at that point, we were looking at having to repeat the trial. It was an 8-year process. I handed the reins over to my second in command, Jennifer Simpson, who ultimately ended up getting it approved last year, her and her team. So great success. But I went on from there to another pharma company that I was on the board of that called Antares Pharma, which made combination products. Auto-injectors and bio-betters, taking existing drugs and making them better by applying a device technology or a transcutaneous delivery technology or a depot formulation, really, truly bio-better. Mm-hmm. And it was quite a factory. It worked with all the big pharma companies and developed very, very innovative and proprietary technology for them and would manufacture the products for them. So I got a real operational education there in development, in CMC, in device, a big device division in Minneapolis, Plymouth, Michigan. And it was really fantastic. I took the job as a director going to CEO because the CEO had left at a very tough moment. There's never a good moment to take a marvelous opportunity. But it was sort of a tag you're it at the board level. Eamonn, you're the best guy and capable guy to do this, and would you consider doing it? And I thought the number 2 there, Bob Apple, was the obvious choice, and I said so. The chairman knew Bob Apple, had raised him from a pup, as he said, and said, "No, I don't think Bob's ready. I love him like a son, but I don't think he's ready." And he knew him better than I did. But I thought to myself, if he's not ready, neither am I. He looks pretty good to me. And so I took the job on the condition that everyone would agree I could develop Bob, and I was quite confident I'd be able to do that. Turned out that was one of the hardest jobs I ever had because when you're CEO, you're the boss. You get to do what you want to do. and you own it. If you're right, you own it. You're wrong, you own it. And life's pretty simple. When you're as a CEO who's developing an imminent number two to take over, you're really always trying to get them to do it and do it in a way you can live with. And Bob's his own man and quite talented fella, and his way of doing it wasn't always what I would have done. I told him, and we're very good friends and colleagues, and but it was really hard to kind of be that mentor, because at the end of the day, in a public company, you're still responsible. So it was a little more stressful than I thought at the time, but it all worked out great. Passed the baton to Bob in 18 months, and he went on to have a great exit with Antares Pharma. It was acquired a couple of years ago for a couple of billion. So it was all good, and very proud of him. He did a great job. So when I left Antares, I said, okay, I'm done with the CEO thing. Enough is enough. I'm going to find something that really is what I want to do without being a public company CEO. So I ended up at the Brigham and Women's Hospital, Harvard Med School. They brought me in to get their translational medicine accelerator off the ground. They had gotten a big endowment to really fund translational medicine accelerator, really going from bench to bedside. And they said, we want an entrepreneur like you who has drug and device development experience to mentor our faculty, teach them how to be entrepreneurs. And I thought, wow, this is cool. I'll have my arms around the whole pipeline of Harvard Medical School. Imagine the faculty at the Brigham. I mean, the best and brightest Nobel laureates, et cetera, et cetera. I said, this is crazy good. And it was absolutely magic. And the faculty was incredibly appreciative, even when my lesson was, your baby's ugly. You need to kill it, fail fast. They were still appreciative that they had learned a whole lot. And I thought I was done. I was really very, very, very happy there. And I got a call from a former colleague who, a research physician, who said, hey, Hobbs, you have to come see my data. I'm curing metastatic prostate cancer. And I said, buddy, I'm literally standing in the hallway at Dana-Farber, and nobody's curing metastatic prostate cancer here. So how the heck are you doing that in Fort Lauderdale, of all places? So he said, well, come down, I'll show you. So I did, and he did. He had developed, or helped to develop, a new method of immunotherapy that was showing incredible promise. And in fact, he had a case series where he was very successfully treating late-stage castrate-resistant prostate cancer patients who were hospice-bound and getting complete responses. Unheard of, including resolution of bone mets. As I peeled that onion, it became clear that he had adapted his case series from another physician who I knew, who had trained him, who had over 400 cases at the time, by the name of Jason Williams, who's the real pioneer of this Sync-T therapy. So went to Jason Williams, did a deep dive on his data. Most of it was in lung and metastatic lung and breast, a combination product of a complex immunotherapeutic with a new device. And I said, wow, this is a massive breakthrough. If it's real, it's anecdotal data, and well, boy, it sure looks real. So I brought in a team to really go through the data, including my old friend Chuck Klink, who was a year behind me at the Air Force Academy. He didn't finish either. He went on to med school in the middle of the Air Force Academy, but in the small world department. And Chuck's a medical oncologist, NCI trained, NCI fellow, and an entrepreneur himself in immuno-oncology. He came in, peeled the onion, and he said, Eamonn, this is the best thing I've ever seen. We need to figure out how to get this to patients. And I said, I'm thrilled to hear that. I'm all in. So I we put together Syncromune and funded it, and got a phase 1 trial in prostate cancer, to the point where we could publish interim data, which we did at the American Association of Cancer Research in April. And I again delivered again at ASCO, one-on-one with some of the leadership, or every leader we could hold still.
Yeah.
And and it's just incredible data. We have an 85% in our evaluable patients, which is 13 out of the 15 in the trial at that time. 85% overall response rate, and 5 complete responses, which included complete resolution of bone mets. In immunotherapy, when you get a deep, complete response like that, it's very often durable, and after 5 years, you can start to call that a cure. Our longest patient out now is well over a year, disease-free, and we couldn't be more thrilled. We have another phase 1 trial with a different drug in metastatic lung and metastatic breast, and we just had I can't talk about that, but we're having great success there too. Really exciting. It's truly the best project I've ever been involved with. It certainly has the potential to be the most impactful, in that there are millions of patients every year who can benefit from this therapy, and it's just magic. I think it's worthwhile to talk about the complexity of the drug device combination, in that it's not just a drug device combination, and those are hard. We all know that. I always say developing drugs is really hard. Developing new innovative devices is really hard. When you combine them, it's really, really hard. It's kind of the Mount Everest of challenges, from a development, a regulatory, from clinical development, everything is just harder. So the juice has got to be worth the squeeze. The first thing to think about drug device combinations they are going to be harder. But if the juice is worth the squeeze, so what? We know how to get these over the finish line. You just need the resources to be able to do that, which can become quite significant. So if the market's big enough and the unmet need is big enough, then it's still very viable. And in this case, that's the case. So SyncT is a combination of a proprietary device that creates an in situ vaccine by doing oncolysis of a small part of one selected tumor. Mm-hmm. We're not trying to ablate the tumor, not trying to kill it. We're just trying to rupture cell membranes in a preponderance of cells in a small part of the tumor to release the cellular contents, which include the all-important antigens, tumor antigens, which the immune system can use to build an immune response to that specific cancer. So it's a personalized vaccine. And into the device, the second thing the device does is provide a conduit or a needle to infuse the drug, which is a multi-target immunotherapeutic comprised of 3 monoclonal antibody checkpoint blockade, and one nucleotide, one oligonucleotide TLR9 agonist. So we are hitting the immunosuppression with multiple targets, which is quite effective and been shown to be quite effective with systemic therapies, but prohibitively toxic, and being highly immunostimulatory at the same time. So the drug is doing a lot of things all at once. But the mechanism of action really is to synchronize, hence the name Syncromune, synchronize the location of the 3 things you need in order to get an activated and properly educated T cell. And that is, you need the antigens, the tumor antigens, you need the immune system components to do their thing and educate a T cell. And then you need the drug necessary to combat up the highly immunosuppressive aspect of what makes cancer, and be highly immunostimulatory to force the interaction to take place when you've synchronized the location. So the antigens, the immune system, and the drug, that's what the infusion does. And the magic can happen in the tumor microenvironment, but we think more likely the infusion washes the antigens and the drug out of the tumor microenvironment into the draining lymphatics, where we all know have the highest concentration of immune system components in our bodies. Mm-hmm. So it's the perfect place to create the synchronization of location. So think about this as it's a brand new device that has absolutely no utility for anything. You don't want to lyse tissue in the body. You want to ablate it and a margin of healthy tissue around it to have a positive effect in focal tumor therapy. So lysing a small part of it doesn't do anything for anybody except in our therapy. Yeah. You want to infuse a drug in relatively high volume into through that conduit that's going to take the lytic debris and wash it out into the draining lymphatics. And then you have a 4-component large molecule co-formulation. Other than that, this is really easy, right?
Real simple, yeah, real simple, real simple to explain, real simple to take to the VC community as well. I'm curious as you're talking about. I mean, scientific challenges aside, you alluded to the business challenges, the regulatory challenges that the company faced. What did you know going in, based on all your previous experience? What did you know going in about how difficult a wall that was going to be to climb, to go out and say, hey, you've never seen this before, but you ought to give us a whole lot of money so we can keep moving this thing forward. And then go to the regulatory authority and say, hey, you've never seen this before, but give us all the green lights, man, because we're doing some special stuff here. What did you know going in, and then what did you learn as you lived it?
Oh, I knew going in that this was going to be one of the greatest challenges, not only that I had faced, but that anybody had faced in drug device combination development. I'm not aware of anybody who's done a multi-component, 4-component large molecule co-formulation of any kind for any reason. So there was that challenge, and at that time when we started the business, there was no precedent for a co-formulation of any two monoclonal antibody checkpoint inhibitors. Now there's a PD-1 LAG-3 co-formulated product that has been FDA approved, so that certainly helps. But back then, no. Yeah. So investors looked at this and they said, "You're doing what with what?" And so we would show them the anecdotal clinical data and say, "Here's the signal that's got us so excited." Yeah. And it's not well-controlled data. It's practice of medicine data, and it's very, very hard to sift through. But look, the complete responses are there, and they've been durable. The pioneer, Jason Williams, Dr. Williams, has patients out over a decade, and this has got us really excited. But all of that aside, we know we need to develop in a very rational and well-controlled way a new drug and device. And we know how hard that is, but we've done this before. We've assembled the best team to do this, and we think the juice is worth the squeeze. The investment community was very incredulous. You just have to keep kissing frogs until finally you get lucky. And we ended up funding the company through high net worth individuals, including ourselves. Without any classic institutional sponsorship, mm-hmm. to get to the data we have today. So it was a unique sort of investment pathway in that we convinced very smart high net worth investors, but they also had the altruistic side of, hey, they've done incredibly well and they wanted to invest in things that could really help people. And this certainly checked those boxes off. And they invested, the institutions or high net worth individuals, it's as much about the jockey as it is the horse. So they invested in the team. Mm-hmm. And Chuck Link and I, are being serial entrepreneurs, had some credibility, and we surrounded ourselves with an extremely credible world-class team that the investors appreciated. And then we brought in an independent board early on. We call it a public company ready board, to further validate that we weren't crazy. We're trying to keep our feet on the ground. And our lead independent director is Doug Watson, the former CEO of Novartis. and he was the chairman of Dendreon, the first immunotherapy company to get approval in prostate cancer. So an incredibly credible, seasoned big pharma executive that added a tremendous amount of adult in the room supervision, as he always does, in a very kind and gentle way. But when the Oracle speaks, we all stop what we're doing and listen for sure.
Yeah.
We also have Norm Schwartz, the chairman and CEO of Bio-Rad, a very, very large medtech company, who's very passionate about cancer therapy development. A public CEO who took the company his parents founded to over 15 billion in market cap, and it's just a sharp dude. And then Jeff Cleland, a serial entrepreneur in biotech, who is a biomedical engineer, MIT trained, and is on the executive committee of Bio. So he brought just a wealth of entrepreneurial experience to the board. And then a physician by the name of Joe Maroon, who's really a legend in big pharma. He's been on many big pharma boards, but also he was the former chair of neurosurgery at UPMC and trained most of the great neurosurgeons in the world. He's up there in age, but looks like a young man and acts like a young man, and has incredible DNA. He's also the team doctor for the Steelers and super passionate about cancers and including the potential for this to ultimately go into GBMs and other brain malignancies. So we have a really crack board, and that helped with the investors a lot. We're right in the middle, actually, of doing a crossover Series A, so which will be institutional and will set us up for an IPO once the market for IPOs reappears, which sooner or later it always does. So we're ready to go. We're audited. We're all that stuff. But there was nothing easy about this project. That's for sure. Funding was tough. The scientific community was incredulous. The data looked too good to be true, which is an irony. When your data is good, people challenge it. When it's too good, they're really challenge it. Like, how could this possibly be? And I had the same reaction when I first saw it.
I was gonna say you as recruited CEO had the same reaction. Now you get your mind wrapped around it. You're sold on it. You decide you're going to invest in it and build a company around it. You've got to face some of that same sort of incredulous reaction from the people who you want on your team. I mean, board aside, when it came time to build the Syncromune team and put a staff together, how did you go about doing that? Given the fact that you've got to have obviously biologics expertise, device. Drug delivery device expertise, and then the combination of those two things, limited pool to fish in, I'm sure.
Indeed.
So how'd you go about getting that team pulled together, and how did you go about overcoming the skepticism that surely, I mean, it's a risk to join any startup. It's a big risk to join this one, right?
It is. No question. The percent of biotechs that make it is small. The vast majority fail due to this or that. So it's a highly risky game, high risk, high reward game, and folks signing up for that have to be cut from that kind of cloth. Have to have that entrepreneurial attitude and preference that they like the adrenaline, so to speak, of being on the edge. Because it is the edge. In reality, we try and mitigate as best we can. Chuck and I founded the company, and we brought our best and brightest from our former lives to the team, and that's incredibly helpful because you already know, they're known entities. You already have the rapport. You have the trust. Their strengths and their developmental needs. So managing them is less of a learning experience and more of a let's just get right to it.
Mm-hmm.
And they, Chuck and I are a known entity to them as well. So you can really hit the ground running. They were all very, very busy doing other things. So getting them took some salesmanship for sure. And as entrepreneurship always does. And but we did assemble a really phenomenal team. And the people you look for, I mean, ideally you would assemble a team where everybody has combination product experience. Everybody has both drug and device experience. And the way I try and describe drug device combination product development is a Venn diagram of three circles. And where they converge, these three circles converge, is the new therapy. Mm-hmm. So drug device combination product is really a new therapy. So the three circles are the drug, the device, and the procedure, the all-important procedure, because if the procedure doesn't go well, the drug and the device aren't going to work. If the device doesn't work, the procedure and the drug are irrelevant, et cetera, et cetera. The drug by itself doesn't work. So it's truly a Venn diagram. So every decision the development team has to make has to be in the context of that Venn diagram, in that you can't just do what's best for the drug. It has to be what's best for the drug, but also works for the device and the procedure. You can't do what's just best for the device. It has to work for the drug and the procedure, et cetera, et cetera. Really challenging. So when you build the team, you need expertise in all three of those circles, plus the convergence, the therapy. So how many of those people exist on the planet? Very few.
Yeah.
So a lot of time you end up with people who are expertise in the drug, but really have very limited, if any, experience in the other two circles. So you have to do a lot of education. And vice versa for the device procedure team. Most device people have procedural experience because of the nature of devices. Very few drug people have procedural experience because of the nature of drug delivery. So having spent decades in combination product company leadership, I have a canned presentation, educational presentation that I give to the team early on that explains how it all comes together and why drug people are what and how they are, and why device people are what and how they are. So to distill it all down, the Venn diagram, and then we go into, well, what's the average very successful drug person? What's their DNA like? Well, they have a lot of degrees. They've got, an MD, PhD, MOUSC. They've got lots of letters after their name, and there's a reason for that. The reason is what's most important in drug development by far is the quality of decision making, because it's not an iterative process. Iteration is too expensive. Mm-hmm. So you really need people who are incredibly well versed in analyzing complex data and distilling it all down to a decision that at least has a good shot of not crashing and burning. Yeah. But again, most biotech companies fail because the hypothesis failed. And that's with brilliant minds that have MD, PhD, MOUSC all over it, making phenomenally good decisions that turned out to not be correct. So tens of millions, if not hundreds of millions of dollars go down the drain because your hypothesis didn't work, and you can't iterate. Now we look at the device world. In the device world, it's not about the quality of decision making. It's about the speed of decision making, because it's all about iteration, because iteration is cheap. It's way cheaper than time. So fail fast, learn, fail fast, keep going. And that is the antithesis of a PhD or an MD or an MD PhD thought pattern. Yeah. It's so when you put these very, very, and if you look at the degrees in the device world, it's typically not a lot of, not as many for sure, advanced degrees, because it's more about do, make those, a lot of engineers, a lot of let's do quick experiments and iterate.
Yeah.
So when you put these very disparate thought patterns together into an organization that has to seamlessly work together in this Venn diagram, it's oil and water. Unless you
Sure.
educate them right away that there's a reason why your colleague across the Venn diagram doesn't think like you do.
Yeah.
So once the if left to themselves, if the drug people would look at the device people and say, you guys are just frenetic. You just run around with no data, just doing crazy stuff, throwing it against the wall, see if it sticks, and then move on, move on. You give me heart palpitations just watching you do your thing. You talk to the device people about the drug people, and they say, you guys are like park statues. I'm surprised pigeons aren't landing on your shoulder. I don't ever see you move. You're just sitting there thinking all the time and not doing anything. But once you educate them that there's a reason for that thinking, because if you took a device mentality and put it in pharmaceutical development or biotech development, you would crash and burn really fast because
Yeah.
you can't iterate. It's tens of millions of dollars every time you change anything. You've got to be right. So and if you take a drug person and put them in the device world, people will lap the field with them because they're not making decisions fast enough. They're not iterating. They're making really high quality, too slow decisions. So there's method to both that is absolutely valid. And respecting that difference is the lesson of the plan, is that respect your colleague, that they need to think like that, and support them in that need to think that way. You don't think that way if you're in the device world looking at the drug world. You don't think that way if you're in the drug world looking at device. But understand it's a thing that needs to be, and it isn't transferable. And it works. They're all smart people. They
Yeah.
go, wow, I never thought about that, but yeah, that makes sense. And yeah, okay, yeah, I'll stop bugging them. The vice folks will stop bugging the drug people about getting, why aren't you guys done?
Would you is it fair to say that as like in your role as president and CEO, that some of the responsibility in setting that tone and achieving that understanding falls on you? Like,
Oh,
I mean, it's
absolutely, yeah,
yeah.
yeah, yeah. Management at its essence is about alignment. And how do you get people aligned? You plan, and then how do you keep people on the plan? You interact, and so as CEO, you're the penultimate manager, and I'm involved in that all day, every day, making sure everybody's aligned, and the
Yeah.
plans are in place, and it's a symphony. You've got to conduct the orchestra because
Yeah.
it's complex music for sure.
Does that complexity and the need to align fall outside the four walls as well? Like, I've got to think that in a complex manufacturing environment like the one you're in, you've got dependence on a little ecosystem of partners. So
Oh,
tell me about that.
absolutely.
Like, how I guess the base question would be, how do the parts come together from a manufacturing perspective?
Yeah, in biotech, the rule is you work with contract development and manufacturer organizations because the technology is so rapidly evolving and so hyper specialized and so expensive for every company to develop internally that it's just impractical. So you're partnered with a very vibrant and broad, deep ecosystem in contract development and manufacturing in biotech. In the device world, it's similar, but more flexibility. Sometimes you can do it internally. Sometimes you can't. But it truly is a lot of managing of very hyper specialized vendors that are doing the work, the very very complex work, and keeping them all aligned because not any one of them has the whole picture. The molecule CMC folks have no idea about devices, nor should they. The device folks have no idea about the CMC. It's coordinating it all and keeping it aligned is what the management team does day in day out. And then we do have internal things that when we can and when it makes sense. On just a regular kind of make buy. Do we internalize certain things? Sure, we do. But vast majority is managing outside specialty vendors.
Mm-hmm. Yeah, we're running a bit short on time here, Eamonn, but you're good for a couple more questions?
I am.
Yeah. All right. A couple more questions, and we're going to need to wrap things up and probably schedule a part 2 and perhaps a part 3 and perhaps a part 4, because you're one of those interviews, Eamonn, who you make the job easy for the interviewer. I ask one question and it's like winding you up and watching you go, which is great. Yeah, but the third part of that Venn diagram, I'm a little bit curious about the procedural part. What's the strategy? What are the plans for sort of clinical procedural application, given that we're talking about, a unique device, a unique combination of therapeutics, a unique mechanism of action, multiple mechanistic elements to the drugs mechanism. So just give me some, I guess, forward-looking statements around what the plan might be there to actually execute in the clinic, let's say beyond approval, beyond commercialization. How do we take this thing that no one's ever, practitioners haven't seen it before either?
Right. No, the procedure is as important as the drug or the device. And in the clinical development program, the procedure is a tremendous aspect that has to be managed very, very carefully. And how we go about doing that is by proctoring every case in our clinical development program to varying degrees. So to get a site up and running in a prostate cancer trial, for instance, we need 3 PIs at every site. We need a urologist, we need an oncologist, and we need an interventional radiologist. And the reason for that is, depending on the approach taken to treat the patient, either intraprostate or treating a met, the urologists are very skilled at treating prostates percutaneously. They do prostate biopsies all the time and brachytherapy and all kinds of things with transrectal ultrasound approach and transperineal access. So it's a home game for them. Once we teach them the procedural aspect of it, the urologist can certainly do that. But if the patient doesn't have a prostate, just has metastatic disease, then the MET has to be selected for each cycle of therapy, and that's typically CT guided, and that falls outside of the typical skill set for a urologist because they just don't have any need to be doing that. So we need the interventional radiologist at that point, who that's a home game for them. They can access anything from anywhere with CT guidance or any other guidance, and doing needle biopsies all the time. So, and then the oncologist is there to manage the complex post-op care that can be needed because of the immunotherapeutics. We're blessed so far in the clinical development programs that the toxicities have been grade 1 and 2, and very, very low toxicities with really high efficacy, which is in itself a paradigm change and a bit unprecedented. So, but the oncologists are really the only ones with the skill set to manage immunotherapy post-op and all the patient care required. So we need those 3 PIs. So for every case, we've got those 3 PIs there, and to varying degrees. So the urologist is there, the interventional radiologist is there, and the oncologist is sort of hovering, over the procedural aspect, make sure they understand what's going on. And in the room, we have our own urologist, interventional radiologist, a med-surg nurse, RT potentially, dual certified, who really is a jack of all trades procedurally. And then we have an engineer who knows the equipment inside out in case there's a problem with the equipment. And of course, the first case, it's a C1 where we have the proctors really helping, very, very, very intimate in instructing step by step by step. So we're trying to get to the see one, do one, teach one scenario of procedural learning in medicine.
Yeah.
So we keep the urologist, the interventional radiologist, and the nurse and the engineer there until the clinical team's basically doing it repeatedly without any real intervention. The urologist and the interventional radiologist then go to another site to get that up and running. But we leave the nurse and the engineer there permanently. They're there for every case as backup. So if anything goes wrong, and there are it's a busy place. These are typically academic medical centers, and the team might change. There may be a new nurse or a new tech or a new clinician who needs help. So there's a reason for our folks to be there. One, to recognize how well the team is trained, and if there's swap outs, to be alert to that and then get whatever we need there. But in clinical development, it's critically important to realize going in that Murphy is a close family relative, and Murphy's Law is going to apply, and what can go wrong will go wrong. So you've got to be there for every last case when there's a procedure involved. You may be you're hoping you're standing in the hallway bored because the clinical team's just crushing it, and that's a great problem to have. But and that happens until it doesn't, and then you better be there and keep things going, because running the clinic, everything in these development programs is best accomplished by doing it right the first time.
Yeah.
Because the second time is incredibly expensive, and the data you've generated poorly is now a cross to bear that you have to overcome with better data. So the best way to prevent or to deal with problems, prevent them from happening by being very, very attentive all the way through. So once we get to commercialization, though, it's and I've done this many times in my career, it's a similar situation, but we're not running clinical trials anymore. These are practices, and it really comes down to see one, do, and teach one. And the good news is that urologists clinically know this technique inside out. I mean, this is a meat and potatoes foundational procedure for them. Same for the interventional radiologist. Same for the oncologist. Managing immunotherapeutic toxicities is their bread and butter. So
yeah,
it should be fine. And the reason there's such a disparate difference there is when you're developing a new therapy, the most important thing is developing the new therapy so patients can ultimately have broad access to it. And and when you're treating a patient, the most important thing is that patient. So it's a very different world when you get out into the commercial world. And if they don't follow the protocol because it's in the best interest of the patient, well, that's the practice of medicine, and that's absolutely appropriate. That's forbidden in clinical development. You got to stick to the protocol or have an amendment to the protocol because of a need. So you're really trying to do what's best to get it to millions of patients, and that can often be a tough decision that the sponsor needs to be a part of.
Yeah, very good. All right, so last question would be piggybacking off of that excellent and detailed clinical illustration that you just painted. What's next as far as clinical progress or milestones for Syncromune?
Yeah, we just announced our next trial IND approval. The FDA approved our trial, so that'll be starting at 5 sites in the U.S. We're planning on first patient in September, and that's in metastatic castrate-resistant prostate cancer. That trial will segue right into a phase 2 trial. So it's a phase 1b/2 trial now, and the agency's given us that go ahead. So we're off to the races. To that phase 2 trial will be registrational intent ultimately. So our clinical development pathways laid out. We've got a we plan to add a basket trial for the drug we're having such great early success with in prostate cancer, the basket trial in other malignancies, other cancers. And this follows the path that Merck and BMS took for Opdivo and Keytruda to expand the indications with a basket trial. And when you see signals, you just go right to continue that trial to registration. So we're on a very well-paved road there. In parallel, we have another drug that we're pursuing for metastatic lung and breast, and that's in phase 1. We're very pleased with the progress there. Haven't announced anything yet. It's early, but once that data is ready, it'll go through a similar path that the prostate cancer did, and hopefully we're ready for next year's ASCO with that drug candidate in lung and breast, if not sooner, and that'll take on its own development pathway. So busy. Yeah,
busy, and we've well ascertained the complexity of this drug device combination business, not for the faint of heart. It's clear to me after spending an hour plus with you now that your energy, enthusiasm, intellect, and experience are making you the right man for the job.
Thank you very much, man.
Yeah, and I have lots more questions for you, so we're going to schedule a part 2. We're going to get you back on.
Wonderful.
We're going to get you back on, but in the meantime, we got to wrap things up for now. And again, I just really appreciate having you on. It's a fascinating space that you're in, and I think there was a lot to be learned here on this episode for our listeners. So thanks for joining me.
My pleasure. Have a great day. Thanks so much.
So that's Syncromune, President and CEO Eamonn Hobbs. I'm Matt Pillar. This is the Business of Biotech. I'd be thrilled if you subscribe to this podcast anywhere you listen to podcasts, and I'd be equally thrilled if you bookmark bioprocessonline.com, where you can actually watch these conversations take place and see our smiling faces. Find that link in the show notes as well. And you'll also find a link to register for the BioExpo Live that was referenced at the beginning of this episode. We'll drop a fresh episode of the Business of Biotech on Monday morning like we always do. And in the meantime, thanks for listening.